Also first, thank you for the invitation and for giving us the chance to talk here. So I'm calling it a Biomolecular Emulator, but let's say that what the host showed before is a little bit more accurate, because we're not really dealing with biomolecules, but we're mostly focusing on proteins. So our goal is essentially to model the function of proteins using deep learning. And as a short introduction, I have prepared an overview slide for what proteins actually are, to give a little bit of refresher. So usually what you see in the context of proteins, or hear in the context of proteins, is the term sequence. And if you look sequences up, you get this garbled mess of letters. And this already tells you a lot about the structure of proteins, because what they are biopolymers. And a polymer is usually a longer chain or network of materials you build from small repeating building blocks. And in case of proteins, the building blocks are indicated by these letters, and these are the amino acids. And usually in biology, we have around 21 amino acids, which form this very diverse set of proteins. They're just being linked together in sequence. And the backbone is usually always the same, and what they differ are these chemical moieties in the side chains. And you might imagine, depending on the order and the combination you combine these amino acids, you can get very different effects, because that's just in 2D. In 3D, they take on a variety of different shapes. And you might have heard about the AlphaFold research, where they were looking at the folding problem. Essentially, the sequence of the protein determines which will be the most stable structure. And you can get a lot of variety, and this is driving a lot of biological processes. But it's even more complicated in reality, because the things can also wobble around. So you don't have a static structure. You have the whole thing in motion, interacting with other things, and usually you can think of this as some kind of probability distribution, and you have certain states which will be more likely, you have certain states which will be less likely, and this is also simplifying it even further, because you don't have one protein in your body, you have many proteins interacting, doing complicated motions, and essentially driving a lot of biology. And maybe one of the nicest examples for this function of proteins is this ATP synthesis. It's essentially a huge complex of different proteins embedded in the membrane of the cell. And along this membrane, you have a protein gradient. And essentially what this enzyme does, it has this rotary motion. So it's like a pump of these protons. And with this energy it obtains from the motion, it's synthesizing something called ADP to ATP. And this is like storing energy for the cell we use somewhere else. And this is really covered by the distribution of states you can have, and by the interactions between these different compounds. And biological or molecular biology students will recognize these kind of networks. So you have really a lot of things can interact, drive different processes, shifting energy from one part to the other, and really these functions are driving most of biology. And that's why we're actually really interested in them. The problem is the knowledge is increasing, but there is there's only a small fraction known of how these things work, because one aspect is that these experiments are extremely hard to carry out. And I'm once using this ATP synthesis as an example, and one experiment where they try to really get a hold of this rotary motion, they synthesized a special version of the protein where they added a filament on this stalk of the rotating pump, and the filament was fluorescent. And because of this, they could essentially monitor how this thing rotates. And you might imagine those experiments are not easy to set up. They are very specific to a system, they require hard work, so it's hard to really scale them up. And that's where we want to to set in. So going from this basic variety, I already teased that, is where we have sequence, structure, and function. We can look at it and see what kind of methods are already available. In sequence, there was a lot of process. So there was this old legacy DNA sequencing, which is now replaced by the modern next generation sequencing methods, which can essentially produce a lot of data and a huge knowledge bases available, which are also actively used in many research avenues. For structure, there were more recent breakthroughs where you initially had the X-ray crystallography, cryo-EM spectroscopy, and now you have the machine learning models like AlphaFold 2, which were a huge breakthrough for the sequence to structure paradigm, which allows us to also scale up this technology and to build knowledge bases. And in the function, it's slightly more complicated because right now there are not really any scalable technologies or knowledge bases. What you have are experiments and simulation techniques like molecular dynamics, which can elucidate small regions of this space, but you have nothing scaling up. And that's essentially where we want to tie in with our research. And how we envisioned this was to have a system set up like this. So essentially, on the one side, as an input to your model, you want a specification of your molecular system and the environment. So the simplest thing, in our case, would, for example, be the sequence. And then we want a model doing something. In the end, what we would like to get out is this distribution of different conformations a protein can take and how it evolves with time optimally. Because based on this, we can essentially derive a lot of different other things. So these distributions, first of all, tell us which kind of structures, so structural motifs we have access to. We can analyze how our systems talk to each other. We can derive thermochemical quantities, like, for example, binding free energies, telling us how likely drugs are to target different parts of the system. And we can also compute kinetic properties like the binding rates, which essentially would give us a lot of access. Unfortunately, it's hard to do. And to make it simple, the first step, we remove this time dependence. So what we now want to predict is, given a sequence, what kind of distribution is the most likely one to get for the associated protein structures. And with this, we still have access to quantities like the structures and the binding energies. We lose kinetic quantities because we don't have time dependence anymore, but it should be enough to, for example, have some impact on drug design or predict the various interactions in these complex networks to get some kind of quantitative analysis. So how do we achieve this? How do we go towards there? And once again, it's complicated, and there are a few things complicating it related to this non-scalable tech I showed before. First of all, the experimental side is it's not really let's say extremely helpful. So you have many different experimental techniques, but they usually do not really give you an overview over this ensemble you want to predict. Because, for example, in crystallography, it's an extremely accurate method, but it's usually one structure frozen in the crystal. So it's just one single state of these many different conformations the system can take. And there's also the question how similar to the actual cell environment this isolated crystal is. The second thing is optical microscopy. There you have a problem with the resolution because you're at the upper diffraction limit usually. There are other spectroscopy methods. They have usually the problem that they do not really directly measure the ensemble, but they measure, let's say, aggregated scores of the ensemble, like, for example, expectation values. So you know the average, but you don't know how the distribution would look like. And recently, there are also some other more promising methods like cryo-EM, which have the potential to give you more information about the distribution itself. So maybe just to mention, so cryo-EM is essentially you take your proteins in a solution in the water, you freeze it extremely fast and then you make a picture of it. So if you freeze it fast enough, you should get something like a distribution of the different states, which are there at room temperature. Problem is it's very noisy and it's sometimes hard to reconstruct the 3D structure. So that are the complications from the experimental side. So we somehow want to leverage this knowledge. There's also problems on the other side, because while there are simulation techniques which you could use to try to get let's say an impression of the function, they can be very expensive to carry out. So for example, one standard tool is molecular dynamic simulation. Molecular dynamics essentially takes the atoms in the system and use some approximate terms to describe the interactions, for example, bonding terms, long-range electrostatics, and then you use these energies to derive forces and you integrate Newton's equation of motion. And essentially what it does is you start with an arrangement of atoms and it tells you how it moves with time. So you can essentially describe the full motion of the protein, but it takes many, many, many steps in order to really describe biologically relevant processes sometimes. So for example, in this Barnard-Barstar system, you have a bound state, unbound state, and for example, if you wanted to simulate this binary event, it will take 50 microseconds in real time, which in simulation time is around 1 year. If you want to simulate the inverse event, which is a little bit more rare, it would take approximately 1 million years GPU time to simulate. Because it happens across 1 hour in real time, and you need many, many, many steps of these approximate simulation methods. And another problem is, in reality, all these systems are quantum, but you are describing them with approximate terms because otherwise you couldn't afford the simulations. So there might be some discrepancy between experiment and the simulations. So how do we get around these experimental limitations and these synthetic data limitations? Well, the plan is essentially to use machine learning and certain, I won't spoil anything, but we use machine learning and to combine certain data techniques, certain training protocols, and combination of an architecture to really make this problem more scalable, to have something like an emulator for this distribution and in turn the function of the systems we're interested in. And the general idea, as I said before, is we want as an input a protein system described by the sequence. We want our machine learning model and it should predict the ensemble. So first question is, how do we represent a protein? The sequence is easy. That's just arrangement of letters, and there are large language model approaches in order to deal with this. It's also what AlphaFold use internally. What's a little bit more involved is how to actually resolve the structure, so the 3D structure in space. And there we actually use, or other people have used, and we also have adapted this property of the proteins, that there are arrangement of very well-conserved quantities in the form of amino acids, because we can just take this, we say, okay, we can coarse grain the whole thing, we just need to know where these side chains are attached. So that will be the C alpha positions. We need to know what amino acid this side chain belongs to. That's the sequence information I mentioned before. And then it will also be nice to know how these side chains are oriented in space. And that we can actually do by just taking these backbone frames and using them as orientation frames as input in the model. So this is one of the most common ways to represent the structures. And now how do we, given this, how do we, for example, get the ensemble? And for that, we actually rely on diffusion models, which many of you might have heard before. It's essentially an extremely nice way to have a generative model of distributions. The idea is that during training, we essentially start from your from your noise-free data distribution. You noise it up. in our case, we use score matching. So what we learn is a score which is essentially tells you how to denoise these different noised pictures. And once you have trained your machine learning model, you can reverse the whole procedure. You can start from extremely noisy state, usually the Gaussian distribution, and apply your model in reverse. And what you will end up with is more and more noise-free. In this case, pictures. In our case, proteins, which will ultimately follow the distribution you plugged in during training. And for example, would like to look like this. So in the beginning, you have very constant Gaussian distribution of atoms, and then during denoising, you see how it falls more and more of the protein. So this is how we get our distribution. We train a diffusion model, and how do we get in the sequence information? Well, what we did mostly is we built on the AlphaFold 2 architecture a lot. So what you essentially see here are two different parts. On the left-hand side, you have the sequence encoding. On the right-hand side, you have the structure encoding and the diffusion model. What the left-hand side does, essentially, you have the sequence as input, you have some pre-processing steps, and then you have many applications of these conformer blocks, which are essentially transformers similar to what you use in the large language modeling. You end up with a representation of your sequence, so essentially giving you a vector for each amino acid in your system, and a vector for each pair of amino acids telling you how information about interaction. And then we take this, we plug it into a score model together with the backbone frame information during training, and then we rely on variant point attention, which essentially is a way to encode the alpha positions on validation frames in a way that's meaningful for machine learning systems. You can think of it a little bit as a fancy graph neural network, because you have transformer modules where essentially the attention is modulated by the distances between the alpha positions, and not just the alpha position, but you have the point count oriented based on how this rotation frame looks like. The important thing for us is essentially we can then get some readout features, which we then use as a score function, and the score function we can plug into this whole denoising diffusion model training. And in the end, we hopefully get out a model which can describe the distribution of proteins. And just for a disclaimer, we actually didn't train the sequence part. We found out that we can actually directly take the AlphaFold 2 sequence embeddings for purposes right now. And with this, I would give a short summary. So, as I said, we recycle many things from AlphaFold 2. There are some key differences. So we train it in the form of a diffusion model to get the distributional information. We use a lot of new data, which we will show a little bit in the following slides. And then in the addition, even given this data, we can mostly get as good as we can get with molecular dynamic simulations, because there's this inherent limit of physicality. So what we also included is additional training losses, which try to get our model closer to experimental predictions, so that we bridge this last gap. And what we came up with in order to achieve this was a two-step training protocol, where in the first step we didn't care about the quantitative distribution. We just wanted a model that approximately gets protein conformations in different states. So you can think of like getting these delta peaks across space, so being as diverse as possible. And then in the second stage, we would use 3D molecular dynamics data to get these distributions a little bit more physical, to fill in the space of protein conformations. And then we also added these additional loss terms in order to really bias it towards experiment. So to give this approximately correct distribution the last shift necessary. And what I will talk about now is this first training stage. where we develop the protocols based on artificial data. So it's mostly related to sampling conformational states. Conformational states is exactly what I showed before. I want this delta peak here, I want another delta peak there, and I don't really care what's in between initially. So we want a model that can generate very diverse, plausible conformations, but we don't care about quantitative structure at all. There we encounter the first difficulty because usually for these applications you use the protein database for training. This protein database is not very diverse in a conformational sense. So what we use as an approach instead, we leverage this AlphaFold DB database, which was synthetically generated by the AlphaFold model as a starting model for training. It has many advantages compared to standard protein database in that, first of all, it contains more data. It contains repeated entries for sequences and very similar sequences, which is good for us because we can associate different structures to very similar sequences. And there is knowledge that is capable of modeling multi-conformations to a certain extent. So in the first step, we curated this data set to have an augmented training procedure. So starting from the 200 million entries, we first of all tried to group everything together where the sequence is similar. So we ended up with around 1 million sequence clusters. And in the second step, within the sequence cluster, we wanted to have those structures that are most diverse, so that the sequence that looks kind of similar matches to very, very different looking 3D structures of the protein. So this was some structure-based clustering, and in the end, we ended up with around 50,000 of these sequence clusters with the different structures, and then we use this in a data augmentation way for training our model. And essentially what we did there is during training time, we chose one of those separate sequences randomly, and then this sequence would map to one of the structure clusters, in which just randomly select one of the structures in the cluster. So essentially what the model learns is, given a sequence, you have a certain number of possible conformations it can map to. And there's also another aspect to this. So how do you figure out this thing actually works? And there are some typical benchmarks for the protein-related tasks, but unfortunately, they're usually more interested in reproducing the static structures. So you don't really have you didn't have good benchmarks for measuring the quality of conformation prediction. So we had to take our domain experts, lock them into a room to come up with new benchmarks. And what we grouped it into are 3 distinct cases of biological function we wanted to test our model for. And the first is called domain motions. It's essentially, as the name suggests, a motion of protein domains. So for example, in this case, you have this upper part which makes a hinge motion and opens up the whole molecule, the whole protein. And these are usually important as part of the functional cycle of proteins. For example, in this case, you have this cyclic di-GMP receptor module, and it's not very surprising, but it modulates essentially the concentration of the cyclic D-GMP protein. Important thing is we have these domain motion cases. Also maybe to mention on the side, in green we have the predictions of the model, in red we have the original reference structures. And on the bottom, this is one of those plots we looked into a lot for the analysis. So what this is a free energy surface. Essentially, the x-axis tells you how close it is to this first conformation. The y-axis tells you how close the structure sample to the model is to the other conformation. And the color thing is essentially a histogram of the distribution of the sample, of the generated samples. But since we like chemistry, it's not really the probability, but it's the logarithm of the distribution, negative logarithm, which gives you an energy. So essentially, the darker it is, the more likely a state will be in this case. And for these, for predicting the conformations, we actually wanted to have samples close to both conformations. In this case, having these two densities here is ideal. And we also set these thresholds in order to really say, yeah, we have covered this conformation. And what's also shown is the model samples many of the intermediate structures, so it learns to interpolate to a certain extent. Other case we wanted to look at are so-called local unfolding events. It's when parts of a protein unfold locally or detach. It's also part of signal pathways. Here, for example, this is this rhomboid intramembran protease. The important thing is you have this monomeric form, essentially extremely clustered, and in the presence of another protein, it can undergo this unfolding event. Essentially, this central purple sheet motif unfolds, and you have just these two helices connected by the strand. And once again, we liked our potential energy surface. This time, we don't have a 2D surface, we have a 1D surface. There's the so-called fraction of native context, which measures the foldedness. The closer to 1 it is, the more you could consider it folded. And this is once again the negative logarithm of the distribution predicted by our model. So we have this extremely folded state here. We have this unfolded state on the other side with much smaller probability. And finally, we also looked at cryptic pockets. It's once again, meaningful name. Cryptic pocket is essentially a new binding pocket that performs upon motion of the protein. So here we have fascin. It's made of two parts essentially, and they can rotate relative to each other, forming this additional pocket which might essentially be a drug binding target. Once again, we can represent it as these two plots. And of course, I alluded to it before, it's these single proteins don't make a benchmark. So we really had a lot of people putting a lot of effort into this, going through literature, looking at the numbers, the literature reports, trying to figure out the structures, curating it in a nice way so that we end up with a suitable set of benchmarks which we really can use to assess the model quality. I won't go into detail for everything. I'm just showing you an overview of the performance. So this is for these three different cases. What I'm showing here is what we call the coverage. So it essentially tells you how many of these benchmark cases reach at least 0.1% coverage of these relevant conformations, so which deposit samples in this vicinity. And we see in the first case, the domain motions we perform quite well. Similarly, in the local unfolding and folding events, where we have success ratios from 70 to 80%. And we also performed quite well on these last cryptic pocket benchmarks for the whole state, which is essentially the state with the ligand bound. We perform less well for the apo states. Here we suspect this is because of how the data distribution looks like. And with this, I would conclude this part of the multi-conformations and hand over to my colleague, which gets to the wibbly wobbly part.
Thanks, Michal.
Oh, okay. Yeah.
So my colleague Michal has just introduced how we pre-train the model. So there we have used the AlphaFold database so that we can basically enable the model to make a prediction of diverse conformations, but that is pretty qualitative capacity. So now I'm going to talk about how we use molecular dynamic simulation data to fine-tune the model. And so molecular dynamic simulation is basically a kind of synthetic approach that we have some empirical force field, and we put the protein atoms in the water and then evolve the Newton's equation, and then we get a trajectory of how those atoms moves. So then basically you can understand it as for each amino acid sequence, we will have a bunch of like say like 10,000 different structures which can be understood to be decorrelated with each other and can be like roughly estimated to be IID samples. And then we have this bunch of data for training. And yeah, so with this, we just fit them into the diffusion model and then do the normal score matching. So here, what we have collected are about 200 millisecond empty simulations. And these have included both the external resource, whatever publicly available, as well as like, you know, we have burned a lot of compute within Microsoft, thousands of GPUs for a couple of months, and then we collected a lot of in-house data, and then we throw them all to the diffusion model and train them. Yeah, so to give you an idea how much are those simulations, we have in total 216 milliseconds, and this is basically more than anything available in the public domain, as far as we know. So yeah, I think we're also gonna release the data in the future. So if you look at the figure at the bottom, here we show how this dataset consists of. So basically we have about 1,000, thousands of different clusters with a relatively short simulation, and we have a small number of sequences with very long simulations. And then we collected them all together. And one example is this cat dataset. So cat domain is basically a systematic coverage of the protein motif space, and we have selected some, you know, basically like a protein domains from this data set and has generated our own simulations in-house. And then we have separated them into training and test and also incorporated all the other empty simulations, and we have fine-tuned the model. And here we show 3 examples from the test set. So those 3 examples, you can see that they have some local conformational change. And in the middle, we show those 2D heat maps, and these are called TICA plots. So to give you a brief idea of what TICA is, you probably know PCA, the principal component analysis. So PCA is like if you have a data matrix and then you can compute the covariance matrix of this data, and then by maximizing the eigenvalue of the covariance matrix, you can actually get the principal component, and then you can project your data onto the principal components and get some low-dimensional distribution, low-dimensional projections of the high-dimensional data. And TICA is a very similar analysis, but instead of doing a covariance matrix, it looks at the time autocorrelation matrix. And basically, briefly speaking, this is the low-dimensional visualization of our high-dimensional protein structure data. And the first column is from the molecular dynamics simulation, so that's the reference. And the second column is from our model. So you can see that we actually get a pretty decent agreement here, because those are all from the test set, which are below a certain similarity threshold from the training set. And you see we managed to capture all the global minimums and also lots of transition states as well. And especially, yeah, so we looked deeply into the specific protein structures, and we see that they actually agree pretty well with our empty reference, and especially capturing those local unfolding characteristics, as you can see denoted, like highlighted in the cyan and magenta colors. On the right, this is a bit small, but basically we have some statistics of the tissue set, and we can achieve a mean absolute error of the Cas domain, Cas domains below 1 kcal per mole. And we also achieve a pretty small percentage of clashes, which are unphysical samples generated by the model. So this is only about 2 to 3%. And we have to notice here that running MT simulation is very, very expensive. It takes a very long time to get converged to equilibrium. And then to get the same level of coverage and the distribution here, our model is 5 orders of magnitude faster because it just directly samples IID after training on the MT simulations. Okay, so here, this is one specific example that we pick up to analysis. So this is called ACE2. And yeah, so for this protein, you can see this. There are 2 alpha helix proteins labeled in magenta color, and they actually they are kind of like the kind of gates that can open and close. And then behind that, there is a catalytic site which can bind the zinc ion. So first we show that so our model can actually sample the opening and closing of these gates, and it's quite similar behavior which is observed in the molecular dynamic simulation. And besides, we also want to show that behind the helix gate, there is this catalytic site that has been very well preserved, even no matter how the conformation is changed. And we have confirmed that from the the RMC plots that our model has very well preserved the catalytic site there. Okay, so having talked about how we fine-tune the model on the MT simulation, then I have to say that the synthetic data itself is not enough, because even though we know that MT simulation can provide us a ton of structures, we know that it is limited in terms of accuracy because we're using empirical force field, and we know that empirical force field cannot reproduce experimental measurements very well. And then also we know that the simulation is not guaranteed to converge because it takes extremely long to actually get to the equilibrium distribution. So it's actually very important to incorporate experimental data for training. And here I will just talk about one example that we actually use to fine-tune the model. But then this approach is also generalizable to or like other types of experimental observables. So here, what we're considering is the so-called protein stability. So we have found there is this science paper published in 2023, which has measured the so-called protein stability for about 1 million proteins. So this is a really high throughput experiment. And what is the protein stability? So basically, for one single protein, at room temperature, it can go through a lot of different conformational changes. And if you look at the statistics, some of them are folded and some of them are unfolded. And then if you look at their probability and the ratio between their probability, you can basically get the free energy difference, this delta G there, which basically quantifies how many folded states versus how many unfolded states are. And then, this is what provided by this experimental group. And basically, the data that we have is this sequence and delta G pairs. So basically, so they provided 1 million entries, and for each entry, there's one sequence, and there is one number, this delta G, associated with this sequence. But then all of them do not have any structures. So then basically, if we have a model that predicts the structure, then we don't have any supervision label. So how can we do that? How can we actually use this data to train a model? So we have basically used two approaches. The first is since there's no structure, then let's just generate structures for them. So we have basically selected a small subset from this 1 million proteins. So it consists of about 20,000 protein sequences, and we have run in-house simulations for them. And we just incorporate those simulations into, like, together with other empty simulation data and just train them. And also with those simulations, we can classify them into folded and unfolded states. But then we know that since they're run from they're dependent on the initial condition, and they are also dependent of the force field. We know that the fold and unfold ratio is not gonna match the experimental measurements from the simulations. So what we do is we classify them, and then we can reweight the fold and unfold states based on the given experimental ratio, and then we use the reweighted simulation data to train the diffusion model. So this is the first approach that we have done. The second approach is that we developed one fine-tuning approach that can basically back-propagate through the denoising process to match the target. And we developed this approach mainly because we have only 20,000 simulations, but we have much more sequence and delta G data, and then we have to find a way to actually train on the rest of them. So how do we do that? So I will first show you how we evaluate the model and then try to make the diffusion model to predict the property. And the fine-tuning process is basically based on this process. So we first start with white noise structure X capital T here, and then we can gradually denoise it with our model until X0, so we get the clean structure. And then we have some reaction coordinates, the so-called fraction of native contact. So it just basically measures how folded the protein is. And then we can classify them into folded and unfolded states, and we count them. And then after the counting, we get the probability. And then we can do the ratio and then get the delta G. And then we can compare them with experimental data. So this is how we do the evaluation. And then it's very natural that you just compute the difference between them and then use that as a loss term. And then since everything is differentiable, then you can basically backpropagate through the denoising process. And we know that this is kind of expensive because it's a very long denoising trajectory. So what we do is we actually found that there is a very nice property in this problem, because the folding and unfolding is actually a pretty global structure of the protein. So you don't really need a very fine, accurate structure at a very small scale. So we realized that you don't need to denoise it all the way to the clean structure. You can already get a very good estimate of the foldedness from a noisy structure. So what we do is we really save a lot of compute and a lot of efforts just by only denoising a few steps and then directly make a prediction of the clean structure and then make a prediction of the foldedness from that. And in this way, we can do all the way the same to the end to predict the property and compute the loss, and then backpropagate back through the diffusion process and use this to fine-tune the model. Okay, so yeah, here are some evaluation results. So this is what we get on the test set of the delta G data set. So this is so the x-axis is from our model predicted delta G, and y-axis is the experimental result. So you can see that we actually get a pretty good error and correlation here. And then we also have a sanity check on the very stable proteins. So those are the ones that we selected from some public database that are known to have very, very large delta G. This means that they should be very, very stable, and if you sample, say, like 1 million different samples, you will probably have one unfolded states, and the rest are all folded states. But of course, here we cannot sample 1 million different structures, and we generated thousands of samples, and we check their foldedness. They're all folded, so this is good. Another thing that we have looked at are those disordered proteins. So our model has never been trained on any disordered proteins, and those are all very dissimilar to our training set. And we checked how the radius of gyration of those structures look like compared with the experiment. And surprisingly, they seem to show like a very strong correlation. So this is a kind of a bit beyond our expectation already. So besides that, we have to say if you have the sequence to a scalar data, then very naturally you're thinking, oh, how about I just train a regression model? I just directly map the sequence to the scalar. Then that's a very nice model already. And yes, it is. But then you don't have any information of how this happens. But like one nice thing from our model is that what happens there. Say if you can see how, like if you want to know how a single mutation happens that changes the sequence slightly, and how the stability changes correspondingly, you can also look at the structures generated by the model, and you can see what actually happens there. So for example, here we show 3 proteins, and the first one, you can see that, so the single mutations are labeled as the ball there, and you can see that upon single mutation, the protein structures has basically partially unfold because of the different reasons. So for example, in the middle one, it was a neutral amino acid replaced with a negatively charged amino acid. So part of the helix got unfolded. And in the last example, it was previously a negatively charged amino acid surrounded by a positively charged amino acid. And then now, upon the mutation, we changed it to a positive charged amino acid. So we would assume the Coulombic interaction actually makes the change, but the model doesn't know about that. The model just has seen all the structures, but then you can see that it successfully predicts how the structural distribution changes and how the terminal unfolds. So yeah, that's all the results that I want to talk about. To briefly summarize, we have presented this protein ensemble generative model called BioEmu. We have developed and introduced a set of new benchmarks, including this more qualitative benchmark that my colleague Michael has introduced about measuring how we cover the diversity in the protein conformations. And we have introduced the MD data benchmarks to measure more quantitatively how well we can match the distributions. And then we have also introduced the experimental data benchmark that we can also measure how the model predicts the protein stability. And we have introduced this pre-training and fine-tuning to unlock the qualitative and quantitative agreements of the conformational distributions step by step, and then resulting in a model that is only a fraction of the cost compared to the empty simulation. Yeah, and then finally we can review the structure mechanisms upon single mutations, and also develop the new methodology to train on the experimental measurements. And yeah, so next steps, I think, we would like to extend the current model towards more capabilities like multiple chains or involving some small molecules, and we would like to introduce more experimental data to fine-tune the model, and we would also love to see the community to use this model on more exciting applications. And okay, yeah, so finally this is our preprint. We're going through the paper peer review revision right now, and we have open sourced our inference code and also opened our model weights. We have also open sourced the benchmark that we have talked about in the presentations. So if you're interested, you're very welcome to take a look and give it a try. And yeah, and this is a big teamwork at Microsoft, and some of the core contributors are also like part-time at free universities, and we have contributions from a lot of excellent interns. And yeah, that's all. Thanks for your attention.
Thank you very much for the insights into your work. We now have like 10 minutes for questions, and we have a microphone, so if you have a question, just raise your hand, and I will give you the microphone. Test.
Okay, first thank you for the amazing talk. So in the start of the talk, you mentioned that the proteins were clustered together into similar groups to reduce them down to, I think, 1.4 million inputs that you had. And I wanted to ask how these clusters are generated, and if it's possible, if you use the model to also use such clusters as inputs.
So this is a problem biologists have spent a lot of time with. There are different sequence alignment methods, which then tell you some similarity measure by which you can cluster. And as a particular code we use is called MM6, which it's very efficient to basically check the similarity for lots of sequences. So in this case, it's just looking at the sequences, checking what is similar to each other, and then using some basic cutoff to group them together into clusters. And in theory, so if I understood correctly, the second question would be whether you can use a mechanism like this directly in the model. In theory, you could. To a certain extent, we are doing something similar with the MSA embedding we have in the beginning, where we use some evolutionary similarity between the sequences. One main reason for not doing the clustering directly inside the model would be efficiency, because you have to do many of these pairwise comparisons, and the larger your data set gets, the more overhead you would have there, because you would need to update the clusters.
Okay, thank you. Just to add, so does this mean that for each cluster you have one representative? Okay.
Yeah, it's essentially what we call the centroid sequence. This would be the representative sequence. The cluster contains slightly different sequences, and all of them will have different structures. But we say it's the sequence is similar enough, you can essentially treat it as the same. So it's cheating in a sense.
Okay, thank you.
More questions? Thanks for the talk. Have you thought about integrating post-translation modifications into the next steps? And how much compute did it take to train the model, and also compared to the molecular dynamics simulations that you pre-computed?
To take that? Okay, to answer the first question, the post-translational modification is something that's quite high up on the list. We didn't show it in the to-dos, but it's intrinsically related to the essentially modeling the ligands, because you also have this atom level modifications to your amino acids. The second question is a little bit harder to answer. It's mainly because we use different resources for the molecular dynamics simulations than for training the model. Because for training the model, you need a certain GPU power in order to really have enough memory and everything. The molecular dynamics simulations you can run on cheaper GPUs. I would say the pure GPU count, I think the molecular dynamics simulations win by 3 orders of magnitude or something.
Yeah.
We had thousands of GPUs occupied with the molecular dynamics simulations, and for the model training it was, let's say, 60 GPUs or something like this.
Yeah.
So for model training you need more powerful GPUs for a shorter time. For the data generation we used a lot of less powerful GPUs over a long time frame.
Thank you also from my side for this talk. I wanted to ask which simulation framework did you use?
Framework?
OpenMM with the Amber force field.
You still have your question?
Hi, so thank you also for my side for a very nice talk. And just to preface my question, maybe, I'm not that deep into proteins and protein structures, but recently I attended an AlphaFold 3 talk, and I remember that they telling like, the people in the audience that they did face some problems with the diffusion model, actually hallucinating some structures or some parts of the protein that in fact hallucinate the structure, but it shouldn't have any structure at all. So my question to you is if you encountered similar problems with using the diffusion model in your architecture, or if this maybe wasn't an issue. So,
I think the target is very different here. I think our model is trying to push the diversity of the conformation. So I think we're really trying to actually generate very different conformations than the very low temperature global minimal in the experimental database. And I think one of the key problems we're trying to solve here is to tell, say, like how do you quantify the distribution of the structures that you're generating, and how many of them are more real, how many of them are less real. So in principle, all of them are possible. Some of them are at a higher energy level, some of them are lower energy level. So they're corresponding to different probability. As long as it's like a physical structure, so it's a like reasonable generation. Like what we are trying to figure out is can we even make it quantitatively accurate? Like actually the generation will correspond to the correct probability distribution. Yeah.
So maybe in that sense you could also what usual for the molecular dynamics data, we had this filter for the clashes for the unphysical structures, and these are typical structures where one part of the protein goes into another part of the protein, so where atoms would overlap or where you don't form the connections you would expect. And in this equilibrium distribution frame, you could see these extremely high energy states, which are somehow similar to a hallucination. But in our case, this was quite a low fraction.
Okay. We have time for one more question. Just you.
So yeah, I have a quick, shorter question: whether how large molecules can you basically simulate or sample with this model, and how large molecules do you use for training it as well? Like how many atoms are we talking about? or like how many
okay.
Protein?
So
how many amino acids?
For we have two sets of data. So we have this augmented data set where we actually do masking over the sequence. And we have the MD data set. And in both cases, we use, so the largest system is MD data set is around 600 600 amino acids, and we also use this window for masking the augmented data set structures. And it's also the one, I would say, where we can still sample with reasonable speed. You can increase it purely from a memory perspective, but I wouldn't recommend it. So I would say around 600 residues.
Okay, and I have a second question. When you sample with your model, you kind of sample like single points on the energy surface, right? Like one after another. In comparison, when you're doing these simulations where you have like a trajectory that is evolving over time, are there any complications or like differences in the data that you get out of it because they are not in a sense like connected between the different conformations?
So it's what we showed in the beginning, you completely lose any time information with the model. So it's IID distributed, which is also why we still get all the distributional quantities out of it, because if you run the MD long enough, you would have the same distribution. What we are not able to describe would be some properties derived from time autocorrelation functions or kinetic quantities. This is also something we want to look at in the future, whether we can recover it. Because, I mean, you also have this information in the MD simulations, and we are not really using it right now.